So my first topic, the variability of the cortical folding pattern, and the big issue, does it mean something? You probably all know that your fingerprint is unique. You can open doors with it or even pay in some places. And it's the same for the cortical folding pattern inside your head. And nobody really knows whether this means something, whether you can forecast your future according to it, like with the hand. And it's the kind of topic I will develop today. And maybe I will begin with something a bit special. I will do a striptease to begin with. I will show you my brain. This is my brain. For those of you who are familiar with brains in 3D, maybe you will realize that I'm a bit weird. But in fact, everybody is weird. If you take a picture of your brain, you will find a place where you're not like in the atlas, like the usual patterns. So basically, the question is whether it means something. Here, it's another colleague of mine. So if you compare both brains, you will see how different they are and how difficult it is to imagine that there's a real way to match one brain to another one, according to the folding pattern. So I will try to discuss this topic, and you will see that it's a very open topic these days. It's important to ask oneself why it's a topic that very few people are addressing. I guess the main reason is that for most of the people, the reason why our cortex are folded, it's just a trick of nature to put more cortex inside your skull without increasing the size of your head, which would be a problem at delivery. And that's it. This is just random folds and doesn't mean anything. We can forget about it. For a long time, this has been, I would say, the paradigm of the neuroimaging field. Basically, everybody has a different brain, but if you align the brain with a template, you can forget about it. So I will just open this black box today for you. I will begin with a simple observation. If you look at these 9 brains, the 3 ones at the bottom are baboons, these ones are chimps, and these ones are humans. When I'm very nasty, I print a chimp and I let it in my office and I ask people whether they think this is an infant or 6 years old or 8 years old, and very few people realize it's not a human. They are very similar. But what is striking there is that baboons, if you look at their folds, they are all the same. Not in every details, but basically the general pattern is exactly the same. And it's disturbing because baboon and macaques have the same property and have been models of neuroscience for a long time. So for most of the people, this story of variability of the folding pattern was not an issue when you used to compare baboons or macaques. For the chimps, it's a bit different. If you look at their frontal lobes, you begin to observe a lot of variability. Temporal lobes and parietal lobes are more similar. And for the humans, if you look again, try to compare this brain. If I let you for a while look 20 seconds about the similarities between this brain, you will get lost. You really need to have a long experience to be familiar with all these patterns and the kind of roadmap you can find behind. So why is it like that? Is there something very special in the humans related to the human nature? Or is it just because the human brain is much bigger, and if you have a bigger brain, you have more fold and it's getting more messy? This is an open issue. Another interesting point is whether we have the most folded brain, because usually we are anthropocentric, so we think we are the best on Earth, so we are the most folded because folded means big brain. It's not the case. If you look at dolphins, you realize that they are much more folded than we are. Maybe they are so superior to us that we did not realize that their cognition was not at our level. But the reality is that there's probably a rule that relates the size of the brain, but also the cortical thickness to these folding patterns. And if you see this plot here, it's a plot from a recent paper, including maybe 50 or 100 different species of mammals, and they all align well if you use a very simple rule related to thickness, cortical surface, and geoification index. So maybe all this is just mechanics, and I'm losing my time trying to understand whether it means something. In fact, the dolphin brain thickness is about less, maybe 1 millimeter, which much less than the humans, which may explain why there's more folds. Still, another interesting point is what is related to genes in the folds. And we'll do a simple game, the kind of game you do with kids, you know, when they have some cards and they have to match pairs of similar cards. This is 4 different brains, but there's 2 pairs of twin brothers in there, monozygotic twins. And we'll just do an experiment. I will just try each of you to try to find a match, and we'll do a pool to see whether the genes are behind or not. So I give you 10 seconds to find the match. which can be horizontal, vertical, or diagonal. So now at least you realize it's difficult. Who is voting for horizontal match? 2? Hope it's not that. Who is voting for the vertical one? Not even 1, and the diagonal one. So the winner is diagonal. I was hiding the shape of the brain because, of course, the twins have the same skull. So the shape of the brain is the same. So it's too easy with that. But if I remove the mask, you see that the global shape of the brains allow a very simple match. Basically, you have 2 flat brains here and 2 globular ones, which are the twin brothers. So there was more vote for this diagonal match, which means that, of course, the genes are there in a way or another. But you've seen that it's not a strong influence. Maybe there's an influence on the earliest folding stuff, but all the secondary and tertiary patterns are different. Still another point. If you go in the field of radiology, usually you will not find people who know how to recognize this fold, give them the names you find in the atlases. It's just useless in the field of medicine. Nobody is really using it, except in a few situations in neurosurgery. And the reason why, it's just if you look at sections like that, it's almost impossible to recognize something with a high level of success. It's very, very difficult because you have very complicated 3D surfaces intersecting section, and you easily get lost. According to the orientation of the brain, it's completely changing the pattern. The people who are really interested, because we are humans, after a few years of training, they can do it. They can recognize stuff in these sections, but it's not easy. So most of them just do not try to use this folding pattern for application. Except I was saying in the field of neurosurgery, because in fact, for instance, in epilepsy surgery, there are abnormal folding pattern in the area that is triggering the seizure. So it's a way to try to do some guess about where to put some electrodes to try to target the origin of the seizure. And my team of computer scientists for 25 years is trying to develop computer vision tool or artificial intelligence tool to be modern to deal with these folding patterns. And the reason why we are so interested in that is the fact that if you look at this timeline, all the yellow brains here are brains corresponding to in utero time. The brain is folding during the last trimester in utero. Here it's highly premature babies. So the point is that if something is disturbing development, some diseases or some genetic problem, it's changing architectures and there's a relationship with the folding. So it's also changing the folding. So what we are trying to do is to find the signature of some weaknesses that are triggered by developmental events. But why it's difficult? Because there's such a variability across the general population that it's not easy to say that something is not normal. But if you look at the following folding dynamics, the brain is still folding further during adolescence and is beginning to unfold at the end of adolescence until late in age. The brain shapes is always changing. Of course, the main patterns appear in utero, but for instance, the fact that you are losing tissue and you're getting older makes the fold open, and it's another stuff that is interesting if you want to take an MRI image and get some information about the status of a given brain. So this computer vision system we have been developing can take an MRI out of the scanner and identify in the brain a lot of structures corresponding to these colors here, what is called sulci by medical doctors. There's about 120 that can be recognized, and we do that by training. We have human experts having labeled a lot of brains by hand, and we train the system to do the same with new brains. I will give you a few ideas about how it's working. I will not go into the details. And why it's interesting, it means that what is very difficult to do by hand, I mean, if you want to do that in a specific brain, it takes hours. Now it's done automatically. So you can test different things. For instance, you can do simple morphometry, measuring the length of the sulcus or the depth of the sulcus, and test whether it's related to some diseases or some behavior, stuff like that. And I will show you a few examples. One of the models we've been developing for the recognition is based on artificial neural network, which are very trendy these days. But it was done in the old time, and it was not deep learning. In fact, it's what I would call wide learning, because in fact there was 500 different artificial neural networks collaborating together. It's a bit like if each of you was trained to be an expert in the recognition of one of these sulci, and after that I put a brain on the table and I ask you to negotiate where you will find your sulci, and there will be some conflicts, and you just find the best labeling fitting the most of you. So basically, the system is taking an MRI out of the scanner, and the first stuff the system is doing is just aligning the MRI with a standard referential, which is used by the community, a bit like if you want to read a book, you don't read it like that, you just read it horizontally. And after that, there's a chain of processing, and the most important one is that the computer is computing a negative mold of the brain. It's this orange stuff you see here. It's something that you see sometimes in the nuts. And after that, this orange negative mold of the brain is cut into pieces. And the last step, the real pattern recognition, is trying to reconstruct all the sulci that are described in the atlases. For instance, I don't know, here, this red one here called central sulcus is split into pieces here, which are the elementary folds from a topological point of view. And the neural networks try to reconstruct all these sulci. And I will just show you how they negotiate. Basically, they try randomly different names, and they try to find a combination of names that is agreeing most of them. I will show you a movie. Here you see a brain, and the color map is telling you locally whether the society of neural network is happy with a given labeling. So the labeling is these colors, the name of the sulci, and it's changed randomly until finding a combination that is okay. And you will see the behavior of the society of neural networks. So for a long time there's negotiation, and you will see that suddenly there's a kind of phase transition at the level of this central sulcus. This is in fact the most stable sulcus in the human brain, the easiest to recognize. And after that, there's a kind of propagation to the rest of the brain. It's funny because it's not something that was coded in the system. It's just something corresponding to what human experts are doing. They just recognize the easiest places, and after that, step by step, they try to identify more variable sulci. I will not say more about these things, just to give you an idea how it's working. So a few examples of applications. Maybe the simplest one to understand, there's been a lot of interest in the field for 20 years in the correlates of handedness. Are left-handed people born with a different brain? What is the origin of left-handedness? Of course, if you do functional MRI, you see right away all kinds of things. But the question there was whether in the morphology of the brain there's some correlates of handedness. So it was the first experiment done with this artificial anatomist. So we took a bunch of students from, I think, Montreal at that time, and we just applied it to recognize all these sulci. And for each sulcus, we compute a simple asymmetry index just to know whether the sulcus is longer in the left or in the right hemisphere. And here you see a population of left-handed people, here of right-handed people. And the color code here is telling you whether the sulcus is bigger in the left hemisphere or in the right hemisphere. And to stay simple, if I focus on the central sulcus I already mentioned, this is the sulcus hosting sensory motor areas. You see that it's yellow in the right-handed people, it's bigger in the left hemisphere, which is well known, of course, that it's the dominant hemisphere in right-handed people, and it's flipped in left-handed people. It's bigger in the right hemisphere. So basically, here the finding is that the sulcus is longer in the dominant hemisphere. So the remaining issue is whether you're born like that, or it's something that is stemming from plasticity. And to answer that, in fact, we did study people who are born left-handed, but were forced to switch hand at school. It was German people, in fact, in the 60s, at a time where you were still forced to switch hand at school. And what we found is that they have the asymmetry of right-handed people. So basically, there is the fact that you spend hours at school writing with the hand that is triggering the plasticity and the development of the synapse, the neuropil in the dominant hemisphere, and is pushing the folding process further. But the real application of this stuff is in psychiatry, all the pathology that are related to development. And I will show you here another example with schizophrenia. Here it's a population of schizophrenic patients who are homogeneous in the sense that they have heavy auditory hallucination. No medicine can solve it. And what is discovered is that there's a couple of sulci that are not as developed as in the general population, as in controls. All the sulci in green here. So you see some that are on both sides, the estes, the superior temporal sulcus, it's the auditory areas. And you see also sulci in the language system, here in Broca, here in Wernicke kind of area. And what is striking is that if you see this functional map here, it's a brain mapping experiment, you see the language system and you see it's the same places. So basically these people who have auditory hallucination have also a kind of atrophy in all the folds related to the language system. We don't know yet whether it's something that is triggered by their symptoms or it's linked to the origin of their diseases, but it's just to show you that these folds that most of the people think are just there random, they embed a lot of information about even behavior. And if you take the same population, if you try to go one step further, you can, for instance, ask them, this voice you hear, do they come from the sky or do they come from inside your head? And they can answer to this question reliably the same way years after years. And we use that question to split the population in two. And we discover once again in the same sulci that one of them was different across the two subpopulations. The superior temporal sulcus in the right hemisphere. There's a place here where the population hearing the voice from the sky has a sulcus deformed throughout the front and the other one throughout the back compared to control. And where once again it's getting interesting and you begin to believe in that here you have activations corresponding to an experiment where you put somebody in the scanner, you make him hear some sounds, either from the right ear or from the left ear, and you ask him in fact to tell you whether the sound is coming from the right ear or the left ear. Basically it's an experiment about the localization system in the brain, and it's exactly this place. Which means that there's a link between the architectures of this area and the symptoms they have. Probably this is completely unrelated with schizophrenia, but it's just to tell you that these cortical folding patterns are a proxy of your architecture, and here it's proven by the fact that different architectures are leading to different symptoms. So the other application of this study of the cortical folds is not about development, but about neurodegenerative diseases. I was telling you, in fact, that years after years, your folds will open, unfortunately, because you are losing some tissue. For most of you here, I guess they are almost completely closed. For me, they are already largely open, I'm afraid. And why is it interesting? It's because when you do MRI of old people, even more people with neurodegenerative disease, there's often a lack of contrast between white and gray matter. Furthermore, this contrast, in fact, is probably changing with age because it's related to myelin and there are some microstructure changes related to aging. So it's never that easy to link cortical sickness or amount of gray matter to diseases. If you look at the contrast between tissue, brain tissues and the CSF, it's a strong one. It's a contrast that is so strong that it's not depending on the acquisition parameters. So you can easily compare acquisition done in a scanner with acquisition done in another scanner. So in fact, it's a great way, in fact, to quantify regional atrophy in the brain. Furthermore, if you think about the atrophy process in the cortex, it's occurring, for instance, in one of the cortical mantle side along a sulcus and not the other one, or it's also occurring in white matter. So if you have tissue disappearing both in the cortical mantle and in the white matter, there's an accumulation of volume disappearance that is integrated in the cortical opening. So at the earliest time of atrophy, you already observe some changes in the width of the fold. So it's really interesting to test whether you can get some clue about the first stages of atrophy in aging and neurodegenerative diseases. So we've been doing that recently with a huge data set, which is the UK Biobank data set, the greatest one in the field these days. So it's 15,000 people. And here, for a few large sulci, you see two charts. The blue one, in fact, is the evolution of the opening of the specific sulcus chosen with age. And each age time, for each age, we have about 200-300 people. And the red one is sickness. So you see that while sickness is decreasing, the sulci are opening. So, of course, it's strongly correlated, but sometimes you see some more information in the opening. For instance, if you look at this one, you see that there are two different slopes. Around 50, 55, there's an acceleration of the opening that may be related to some neurodegenerative diseases behind. You all know that there's a large proportion of the people in the aging population on the path of Alzheimer's disease, for instance. So basically, it's another biomarker of interest. And we've been testing whether it's related to genes. So for instance, here it's a heritability study using the Human Connectome project. It's about 1,000 subjects. About 2/3 of them are twins. So you can test for heritability. And I was telling you before with this simple game that the genes are behind the cortical folding patterns, but it's not so strong. For the largest one, you see here in red that the heritability of the opening of the fold is high. So that there's really all kind of clue there telling you that this is not just random stuff. It's really related to all kind of things, aging, atrophy, genes. I'm just showing that to show you that this is like other biomarker you can quantify from MRI. And do we find some stuff related, for instance, to dementia? Here it's a simple experiment with a French cohort of 2,000 subjects who are at risk of Alzheimer's disease or other related dementia. And if you correlate, in fact, the opening of all these sulci with a memory test, you see that in some of the places, in most of the places, there's a strong correlation, significant. And there are some places where it's really, really strong. For instance, the two superior temporal sulci. It's something that is coming out from all kinds of experiments with various diseases. It's like if it's a place where you have the beginning of the atrophy triggered by cognitive difficulties. So basically, this morphometry of the sulci is used not only for development, but also for study of degeneration. And now I will switch to something I think more interesting. I will teach you how to read the lines in your hands. I'm sure everybody here is interested in that. And maybe because some of you, it's a joke. Maybe some of you have already tried with the lifeline. We'll try with two other ones. I guess most of you don't know. In fact, there's two other lines here in your hand. One is the line of head. It's supposed to be related to your cognitive capacities. And the other one is the line of the heart, which is related to your emotion. And I would like that each of you try to find it in one of your hands, maybe the same as here, to simplify. Normally, men and women have to choose a different hand. Is it easy for everybody? Or is it difficult for some of you? And can you tell us why it's difficult?
Who knows that they're there? I mean, I have no idea what I'm seeing. For these weaker ones, at least.
For instance, in my hand, for the people who know at the first rank, do you see these two lines in my hand?
Only one.
I have only one. So basically, the people trying to read your line have the same problem as I have when I try to recognize the sulci. Why it's getting disturbing is that if you take Down syndrome, more than half of the Down syndrome people have this pattern I have, only one line. I'm not the only one, you see. And why is it like that? People are not aware most of the time. It's because the genes that are behind the development of the hand are the same as the one behind the development of the brain. So genes acting on the brain act on the hand too. But it's just to tell you that what we are really interested in is try to decipher these patterns because some of them are really related to problem in development. But it's not that there will be a folding pattern that is the signature of autism. It's just the signature of some weaknesses. And for instance, this pattern I have, 10% of the population have it in one hand, 5% in the two hands. So it's not that there's 5% Down syndrome on Earth. So it's just to make you realize what is behind this quest. And I was mentioning at the beginning that, in fact, we don't really know what is causing the folding. It seems crazy, but in fact, the research on this topic has really begun 10 years ago. And now there's really great stuff. For instance, this is a simulation that has been proposed by a mathematician from the MIT 5 years ago. they have found an equation, a very simple one, that is modeling the fact that the gray matter is growing faster than the white matter, and it's triggering falls. And if you look at that, it's looking like crazy as a real brain. So it seems they found the equation behind the folding. But the folds, they are not at the right place. You don't see the central sulcus. So there must be something missing. And surprisingly, this thing missing is occurring from the world of biology, from the world of people working with the ferret brain. I'm afraid with the contrast you don't see a lot, but here it is a ferret brain, and they use a tracer of the neurons during the proliferation around the ventricles. And you see that there are some places where there's much more neurons than elsewhere. In fact, there are some places where the proliferators create maybe 100 times more neurons than in other places. And it's exactly these places that become the gyri. And there must be a kind of protomap like that in humans that is leading to the main folding patterns. So the idea behind what we are trying to do for a few years now, we try to forget about the description of the old anatomists, that we are just there with a few brains on the table, and they could not do a lot out of that, so they just propose a roadmap that is what it is. But there's no dictionary of all these folding patterns that you can find in the general population or in pathology. So the idea is that, for instance, if you take the superior temporal sulcus, you find in the general population 3 patterns. One where the sulcus is long, elongated, without interruption. Another family where there's a strong interruption at the back, and the posterior part is above the anterior part. And you have another category of the population where the anterior part is above the posterior part. And we think that this is just because the protomaps are a bit different in these different groups. So the idea is that if you find one of these patterns, you can guess something about the earliest process of development. And here it's a chart to make you understand more the idea. It's coming from, maybe some of you know Turing equation. It's a very simple non-linear system leading to patterns. What is nice with this system is that there's only 2 parameters. So if you play with the 2 parameters and you try to simulate a folding surface, you have some parameter regions where you get folds looking like folds in a real brain, some places where there's no folds, and some where there's too many folds, a bit like some genetic disease you find in humans. And the idea is that there are some areas leading to a specific pattern for this sulcus. And there are different areas like that. And if you go away from these usual areas, you find some pathology because there was something weird in the protomap during development. So it's really what we are trying to do now. And still another analogy to make you understand the story. Of course, you all agree on the fact that in humans, there's an architecture of the hand with 5 fingers. But you find rare cases where the hands are not like that. Sometimes it's not a big deal. If you have 4 fingers like that, it's fine. The hand is very resilient. And the brain too. Maybe it's the explanation why, even if I have this line, I'm not completely healed. But of course, there are some very bad hands here that cannot be used in the normal way. And it's exactly the kind of stuff we would like to discover in the brains of people who are at risk of developing some psychiatric syndromes. Of course, you can look at the brains yourself, and we even have great tools to do massive visualization of a lot of brains at the same time, but it's very, very difficult. I mean, we are not born to do these kind of things. I mean, brains folding are so complicated. If you look at a fold, and after you try to think whether the pattern you see in this fold is the same as in another one, you right away forget about the first one. And if you want to compare 100 controllers with 100 people, I don't know, with schizophrenia, it's a nightmare. But some people are really motivated. And I have some colleagues in Paris who found a very striking pattern. They are working on epilepsy, on motor epilepsy triggered by motor areas, which is easy to stratify. And they discover almost by chance using these tools that more than half of the people with this specific kind of epilepsy that is caused by a dysplasia in the motor area, some neurons did not finish the migration, they found this pattern, which is very ironical because this is the power button of the computers. And it's very rare in the general population, less than 1%. So it's the kind of stuff we are trying to discover, but with a more systematic approach based on machine learning. Basically, you were aware that millions of people on Earth now have an MRI of their brain. So it's exactly the kind of situation where you can hope machine learning can find something. So what the program we are developing for 5 years now is maybe the program we would develop if we are people arriving on Earth in 10,000 years from now. no humans on Earth, we are all dead, don't know why. And they found some books written in Chinese. And they guess it's a kind of writing, but they don't know what it means. The first stuff they would have to do is to try to do a dictionary of ideograms. I was thinking about Chinese writing, because with alphabet it's not the same story. So really we are trying to find the ideograms of the cortical folding. And we don't know exactly how many we need, but machine learning is just born to do this kind of thing. So, and we are not in the situation of the old anatomists. We are just looking at brains without knowing anything about the brain functions. Now, for instance, we could use data, for instance, from the Human Connectome project, where for the 1,000 brains there, they have individual parcellation, mainly based on architectures. There could be a lot of discussion about that, especially here, because it's not architectony, it's something else. But basically, with this, to drive the machine learning stuff, we could try to find a dictionary of patterns that are related to architectures, which have more chance to be meaningful for what we want to do. So this is the kind of program we have in the HBP. And I will just give you an example of what it can lead to. I will follow my red line, which is the central sulcus, the most stable one in the brain. It's the reason why we like to play with it, because at least we can extract the central sulcus for the brains, from the brains. It's this red shape here. And you see that this is not just a surface, there's a kind of deformation in the middle. And we know since the advent of fMRI that this deformation is normally, in fact, the hand area. It's the place triggering your hand motion. But if you look at all these central sulci coming from maybe 30 different subjects, you see that their shape is very variable, and sometimes there's even several deformations, so it's unclear what it means. If you use simple machine learning tools, for instance, what is called manifold learning, you just ask the computer to order the sulci according to their shape, so that two sulci with the same shape or similar shapes are at similar places on these axes. You get that without saying anything more. And you see that on the left of these axes, you have sulci with a big deformation that are in the middle of the folds. And if you move towards the right, you get more complicated shapes. 2, 3 ondulation, it's the finding of the computer. And we did that with a lot of data sets, with thousands of brains, you always get that. You see here with 1,000 brains, you guess, at least for the people on the first rank, a kind of line here. In fact, it's this deformation here moving up. Here it's in the middle of the sulcus, and it's moving up here. And if you want to have a better understanding of the shape that is coded by these axes, we can do some local averages. The kind of stuff people do in the stock market if they want to get nice chart. And you see that the average sulcus here has really a shape with a big deformation in the middle. And the one on the right side, a shape with 2 deformations. And if you do fMRI, you can check that this deformation that is here at the top is really the hand area. It's where you get the activations here. And where it's getting interesting is that if you do an experiment with language, there's a premotor activation in language, which is probably the area in charge of articulation and this kind of stuff. This is the blue area here, and it's moving. Here, it's not in the central sulcus, it's in the precentral sulcus that is in the front that we do not see here, while at the other extremity of the axis, it's in the central sulcus. It means that the shape of the sulcus allows you to guess the architecture here, but if you follow the standard paradigm of the field, you just align the sulci, these activations are not aligned across the people if they have different shapes. So basically, all this is going toward the direction that there's really something to be deciphered there, and machine learning can do it. If you take the experiment with left-handed people, in fact, in the left hemisphere, the dominant for right-handed people, they have a majority of 2 knobs in their sulci. Left-handed people, they are here. And if you take the people switching from left hand to right hand at school, the ones who have a parent that is left-handed, so who is left-handed probably because of genetics reason, in fact, they stay here, even if they have the asymmetry of right-handed people. And the other one move a little bit. So it's really a way, all these analysis, this morphometry of the folds to get some stuff about architecture. And to finish with the folding stuff, a last experiment just to show you that you can really guess some weakness of the brain. It's an experiment with a database that is very special. It includes controls, people who have amputation of one hand occurring at adult age, and people who are born with a missing hand, only one. And if you play the same game, you ask the computer to do the manifold learning and just one dimension to keep things simple, you don't get the usual organization that you get in the general population, you get this. You get the fact that the main variations in this population is going from a flat sulcus to a sulcus with the usual knob pattern. And the people who create that, it's not the people who get amputation, it's the people who are born without a hand. So in fact, if you're born without a hand, kind of intuitively, you don't get the development of the hand knob. If you're born with a hand, you have your hand knob, and if your hand disappears, the knob is staying like that. Just to say that all these shapes are really proxy of architecture, and they are stable even when such huge event as an amputation occur. And the other interesting issue we are trying to work on, using a large database of highly premature babies that now are safe even if they are born at 6 months of gestation. Their brains are almost not folded, and we can follow them in time in the context of clinical research. So now we try to guess whether features like, for instance, the position of the knob in the central sulcus can be predicted as early as 6 months of age. Because of course, if it's related to handedness and can be predicted at 6 months of age, it means that maybe handedness can be predicted at that time, which is one of the models of people trying to look at the motion of the fetus in utero. So it's the kind of stuff we are doing. And of course, you can also do comparative studies, phylogeny, stuff like that. And I will stop there, just finish with a warning. What I'm doing is very, very close to phrenology, but I'm not the only one. Even fMRI is not that far from it. Phrenology had more success than people are aware of. For people who don't know that, 200 years ago, people were just supposing that the size of the bump of your skull can help you to get something about the development of your brain. It was just bullshit. I mean, the vision of the organization of the brain was not the right one. But of course, it was trendy. They even developed some phrenometer, not as successful as MRI. But people could do all kinds of stuff with morphometry. For instance, maybe you're aware of what people have been doing with Einstein brain. Maybe you don't know the story. Einstein brain had disappeared after his death. His medical doctor just steal it and disappear for 30, 40 years. And finally it was released, and people now try to look in the folding patterns of Einstein to see whether they can find the folding pattern of the genius and stuff like that. Of course, it's going much too far, so we have to be aware that this is not the game. What they found is that Einstein has 2 right hemispheres, of course. So now we'll switch to the fiber bundle. The reason why I'm working on the fiber bundle, it's because they are not known, because what could be used before using dissection or tracers could not be applied to humans for this stuff. So it's only the new technology that allows us to have a window on these U fiber bundles. What I'm calling U fiber bundles are short bundles connecting different parts of the cortex separated by 1 or 2 folds. And if you do not control for the variability of the folds, it's very difficult to map them. This is done in the context of the Human Brain Project, and as Marcus was explaining you before, PLI is central to the HBP program on connectivity, but PLI is something you can just do on a few brains, and diffusion imaging can be done on a lot of brains, so it's a way to study also the variability across people. I guess some of you are not familiar with diffusion, so I will just take a few slides to make a few things clear about this modality. It's not a modality where you are tracing axons, so you can do things as incredible as this technique that has been developed with rodents, where they have a fluorescence in the neurons, and each neuron has a different color, so you can do incredible stuff. Diffusion imaging is based on statistics. It's based on the fact that the cellular organization is disturbing the random motion of water in the brain, and you can quantify that with MRI. So if there's a lot, hundreds or thousands of axons in one specific direction locally, you can guess it. But you cannot follow, and you will never be following a single axon. With PLI, you can follow one axon, which is crazy, but they can do it. So I will not go into MRI and this complicated stuff. I just try to find metaphor for you to feel a bit better what it means. And maybe the best metaphor I know is a metaphor with the road traffic. Imagine that you're using a good camera, not a cell phone, and you're in a helicopter above a city, and you're doing a picture of a city at night with a long pause, a long acquisition window, and you get this kind of pictures. If you're above a highway, you will get this kind of stuff, and you will easily guess that there's a big highway there. It's not exactly what we do with diffusion, but it's the same spirit. If there's a big pathway with a lot of axons, we'll get a strong distortion of the random motion of water, and we'll guess there's a strong pathway. But there are some places in cities and in highways much more complicated. For instance, this kind of complicated crossroad. And according to your resolution, you can decipher or not this crossroad. And unfortunately, with diffusion imaging, one voxel is just that. So sometimes, with the motion of water in one voxel, we have to guess that there's all this crossroad, which is very difficult. And it's where maybe PLI could go much better. Not because of the way it's dealing with crossroad, but because the resolution may be 1 time better. What's happening in pathology for diffusion imaging is crazily complicated, so I will not speak about it today. And basically, if you think about white matter and a cartoon organization where just a few cylindric a few cylindric axons. Water can be inside the axon. I don't know what's happening there. Water can be inside the axon or outside the axon. But whether it is inside or outside, okay, you memorize it or somebody take a pictures and after I let it go further. But basically, if you're inside the axon, you can move freely in the direction of the axon, and the other direction, you bump into the wall of the axons, so the mobility is lower. Outside the axon, it's more tortuous trajectory, but it's similar problem. So it means that the water during a given amount of time is moving further in the direction of the axons. So if you imagine that you let, for instance, you manage to color, to label some water molecules at a specific place in the middle of a voxel, and you wait for a while, if you're in a big pack of axons all in the same direction, you will, after a while, the water molecules will be elongated, the cloud will be elongated like that. In the liquid in CSF, it's isotropic, there's no perturbation, so the water molecule will go further and in all the directions. In the gray matter, it's a very complicated organization, so at first glance, it's also going in all the directions, but not going as far. And if there's crossing, ideally, you would see something like that, water moving in two specific directions corresponding to two bundle crossing in the voxel. The reality is much, much more complicated than that, and if you look, for instance, at this nice drawing of axons, myelin sheet, and all this stuff, there's a lot of compartment, and water is doing all kind of stuff. Furthermore, all these membranes are permeable, so waters can go in and out with different timing. So it's very, very complicated. So unfortunately for engineers, it's not that easy even to guess that there's a crossing as simple as this one. But even with that, each year the technology is improving. For a long time, the idea was that if you do a few images to measure the mobility of water in a few directions, if you're in a big pathway, you can guess the direction of the pathway. Maybe let's do a think experiment. Let's imagine that there's somebody on Mars who has a machine that is focused on this room and that can measure, in fact, motion of some entities living in this room in a few directions of space. With just 2-3 directions of space, if the machine is registering what's happening here for a few weeks, people will guess that there's a direction where you have more motion, which is the direction of the table, because you cannot cross the table, and so you just move that direction. This is basically what we are doing with diffusion, and you don't need a lot of direction to deal with a simple geometry as this room or as a big pathway. But if there's a crossing, you need much more measurement. So the big move of the field during the last 20 years has been to move from a 6-direction measurement that was sufficient to assess the direction of the fiber in a big spaceway to very complex measurement with hundreds of directions that allow to disentangle all this stuff. And I will not discuss more about it, I will just show you pictures. Basically now we have images where in each small voxel of the brain you have another 3D image that is a kind of summary of the mobility of water in this voxel, and you have extended mobility in a couple of directions that are supposed to be the direction of the fibers. And once you have this kind of information, namely in each voxel a quantification of the few directions where there's a lot of axons, you can just step by step follow this direction to build trajectories that are corresponding to putative bundles. The only problem with this approach is that it's very sensitive to noise and to problems in the estimation. Imagine that I'm just traveling along a bundle. If there's only one place where the direction is wrong, I will fork towards something that is not a real bundle, and I will miss the real one. So these days we know that for the big pathways it's working fine, because this has been validated by all what we know from dissection and using other technology like PLI. For smaller bundles, it's still completely open issue, but a lot of people are trying to observe what we have in these, what we call tractograms. So it's millions of putative trajectories like that, that we try to disentangle, and you will see that we can find in there the main pathways that have been described hundreds of years ago using dissections. And now what we are trying to do in my team and in other teams around the world, we are trying to map these short bundles that are not known, but are similar across people. Of course, the simple stuff to play with this kind of spaghetti plate is, okay, you draw two regions of interest and you just get the fibers that connect these two regions of interest. Is the way people have been playing at the beginning with these tractograms to do the first atlases of the bundles, the long pathways, for instance, this one. For instance, the simplest way to get, I don't know, arcuate bundle is just to draw a region of interest in Broca's area and one in Wernicke area, and you get all the fibers, the putative fibers, it's not real fibers, connected these two areas, and you will get arcuate fasciculus. And so what we are trying to do with the U-fiber bundle is that we are taking a lot of brains, hundreds, thousands of brains, we align them in different ways. This is a completely complex issue because of this folding stuff. And after that, we just try to find the putative fibers that are similar across a lot of brains. And we just decide that this may be a real fiber bundle, and we just put it in one of these atlases that are really putative these days and not validated. And I will not explain the algorithms behind, but we give you a few clues about the simple idea behind. If you have two fiber trajectories, you can just compute a distance between them. For instance, the simplest idea is that, let's say you put 10 points along the first fibers, 10 points along the second fibers, and you just compute the pairwise distance, and you do the average distance or the max distance. And if you use this kind of distance, you can cluster the fibers with similar trajectories, either inside a specific brain or across brains. So basically, first you have, for one specific brain, millions of fibers. You regroup the fibers with similar trajectories and you will get a few thousand fasciculus. And after that, you compare these fasciculus across the brain, and you will get cluster of similar fasciculus that will correspond to bundles. And if you play that game, you will get the long bundles that have been described in the old time, but also a lot of short bundles that have not been described. We have to create some names for them and try to validate them. And once you have this kind of atlas, you can easily project it on any new brains, once again using this simple distance ID. For instance, if you take a fiber in a new brain, you will just link it to the bundle of the atlas with the smallest distance using a threshold to get close to the bundles. So this kind of approach has been developed in a lot of places around the world and is leading to this kind of atlases where we recognize the main pathways of the old time, and now we have models of their variability across people. And if you apply it on shorter bundles, you get these new atlases with short U-fiber bundles. For instance, this is one of the examples we got last year where we have the 100 biggest U-fiber bundles. And this has been inferred from one database and can now be applied on other databases just to try to see whether it's meaning something. And of course, we have to validate these atlases with all kinds of postmortem techniques, including PLI, but we have also other strategies. For instance, we have an old dissection strategy. Maybe you don't know this strategy. It's called Klingler method. If you take a brain and you put it in the freezer for a while, and after that you take it out of the freezer, some of the weak connection between bundle break, and it's like revealing, in fact, the fiber bundles. After that, if you take a scalpel, you can improve the nice pictures. I mean, it's still a discussed technology, but it's really interesting to validate whether the trajectory we find from diffusion exists in dead brain. Another kind of validation is trying to find whether this organization has some link with pathology. And my colleagues, psychiatrists in our lab, just applied this atlas to study a large database of autistic people. And we were amazed that they discovered that a lot of these bundles, in fact, to deal with multiple testing, they were just doing a PCA to have a 3 group of bundles. And one of these group of bundles is strongly related with autism, and also, which is even more important, strongly related to a lot of the scales that are used to define autism from clinics. It's really a surprise for us. It's working too well, but this is the result we just got. So it seems it's a nice way to organize the white matter and to quantify a diffusion signal inside it. So for this Klingler method, the problem is that, of course, it's done with dissection. It's very difficult to quantify, to compare, in fact, what you get from a dissection with what you get from MRI. So our colleagues in Tours have been developing a robotized way to follow, in fact, the dissections, to do pictures from time to time, to have at the end a 3D image of, in fact, of the bundles, how it looks like during the dissection. That can be compared to the result of diffusion tractography from an MRI that was done before the dissection. So it's complicated technology, but it's improving year after year, and it will be a way to kind of validate these atlases with not only one brain from PLI, but with a series of brain. And my colleagues have, in fact, 100 people who accepted to give their brain to the study. So we even have good quality MRI from the people while they are alive. They are between 85 and 90, and after 5 years, only one is dead. But we are waiting. And the third technology is diffusion. In fact, with postmortem brain, you can push the resolution of diffusion imaging. And if you remember my first explanation, with a better resolution, you can disentangle some of the crossing. So we are in the process, in the context of the Human Brain Project, of doing an acquisition of a human brain postmortem with 200 micron resolution. And it's done using a very strong magnet, a magnet that has been dedicated to rodents, which has a very, very strong gradient, something that you cannot do with clinical machine. The brain has been cut into pieces, and for each piece, in fact, we let the piece in the magnet for a week to push the resolution as far as possible. And I hope that in 6 months from now, we will get one full hemisphere with this 200 micron resolution. Also to do comparison with PLI, but for this specific brain, we will have a way to check one by one whether these fiber bundles we found in alive people is also there. And of course, the link with PLI will be important, but the reason why we are really fighting for years now with Marcus is that we would like to have acquisition with the same specimen, which is really, really tricky. And the bundles are appearing year after year, and the most recent atlas we have has now 300 different short bundles. Here you see them relative to the folding patterns of the average ICBM 152, which is really, really comfortable to understand where they are. So it's unclear whether we have 50% false positive there, or 20% or 80%, but this is really a technology that will for sure develop in the future, just to get more out of diffusion MRI than what you can get with the most standard way, like TBSS for the people who know the field. And I think I will stop there. Thank you.